Working? Is my mic working? Yes, okay. Great. So I was just going to talk a little bit about what's currently going on in the clinical trials world. You probably have heard some of the recent press releases, and for some of that I will defer to people who were at the conversations in DC and in some of the meetings, so don't escape. don't leave. Okay. Because there's a lot happening in the world of clinical research. So I just wanted to go over a little bit about what clinical research is and what the different types are, how all of them are important. So one aspect of clinical research is the part that people don't think of as sexy because it isn't a treatment trial, but these are the natural history studies. But I need to emphasize just how incredibly important they are. A natural history study is basically repeated observations over time, over multiple visits at a particular measure. And so this is one way that we can look at what things change over time, how they change, when they change, how fast do they change, how do we measure the change, is it reproducible? So if I measure it or another doctor measures it, do we come up with the same answer? And then the next question is that change a meaningful change? What does it mean to your everyday life? And can we use those changes to predict the future? And ultimately, if we don't have this information, we cannot design a useful clinical trial. Sometimes clinical trials fail, not because the drug fails, but the design of the trial has failed. And so we want to do everything possible to prevent that from happening so that every trial that you participate in has the best chance of demonstrating whether there's a true effect or not of a treatment. So one way of helping design better trials is to find biomarkers. And so biomarkers are, quote, a measurable indicator of the severity or presence of some disease state. And so one biomarker you're probably already familiar with clinically is CK, or creatine kinase. It's also called creatine phosphokinase, or CPK. And it is part of what gets measured at the diagnostic aspect of Duchenne. But it's also not unique, so it's not a specific marker. It doesn't accurately reflect the severity of this dystrophinopathy. It doesn't predict well disease progression, and it varies greatly from day to day. So overall, this turns out to be not a very great biomarker. So we need better ones. And you've probably heard in the media some of the debates about other biomarkers, such as looking at dystrophin expression. So to look at dystrophin expression, we would have to do a muscle biopsy, which is part of many of the clinical trial designs currently. But once we have that muscle biopsy, how do we look at it? How do we define how much dystrophin is in that sample? And that can be a much more difficult question than one might think initially. But what we want is a biomarker that's a surrogate outcome measure in a clinical trial that helps predict disease progression. And this allows us to design better clinical trials of shorter duration, and hopefully we can enroll fewer patients and be able to have a significant answer rather than having to wait years and years in order to determine an outcome. So one of the papers that came out in this past year was a really lovely paper looking at MRI scans of muscles. So one possibility is, could we be scanning the muscle instead of biopsying it in order to look for change? And they compared these MRI scans to the 6-minute walk test, and they repeated the MRI at 3 months and at 6 months. And it was very interesting is that if you quantitate volumes and signals in muscles, is that they were able to detect subtle changes in as short as 3 months. And also importantly, they were able to see changes that were trackable in certain muscles in younger boys. So as Tina was describing, over the course of one's lifetime experiencing Duchenne's, when you're a younger child, you're growing, you're gaining skills, you're gaining strength, and people are doing better, and there's an upslope. And then over time, things change, and then there is more progression of weakness. And so it's been quite a challenge to find an outcome measure in the younger age groups. And so I think that this is a prospect to be a promising measure. And then the next part is there's a huge amount of literature on this, and I didn't go over all of it, involving everything from mice to cell culture to zebrafish to golden retrievers, I believe, of preclinical trials. And so basically, this is where the large amount of research is going on. It takes years to develop a drug with these models. You need to define what you're looking for. Are we looking at dystrophin expression? Are we looking at inflammation? Are we looking at fibrosis? Are we looking at heart function? And then the question is how are we going to look for this? And this is all of what constitutes a model of disease. And once you have this model, you can screen for potential drugs. You want to show that it's a robust method, a robust drug, by showing that it works in more than one model. For example, does it work in cultured cells and work in mice? And then once you found a drug that has potential, you need to figure out what dose is, potentially optimal. How does it circulate in the body? How do you administer it? Is it a tablet? Is it an injection? Is it a virus vector? And then once you give it to an individual, where does it go? Does it get to where you want it to be, in the muscles, in the heart? And then how does your body get rid of it? Is it metabolized by your liver? Does it go out your kidneys and your urine? Where does it go? And then important questions about how much is too much, and is it bad? And so toxicity is an important thing to look at in animal models, because you wouldn't want to give this to a child if it's going to harm them over the long term. And so some of the important questions that are raised are, is there potential for carcinogenicity, which is, can it cause cancer? And embryogenesis, meaning if someone were to father a child or be exposed to it as a mother giving the medicine to a child, by handling it, is there any risk to unborn fetuses? And these are all things that the FDA is going to want to know. So phase 1 is generally the one that has the least published on it and is least heard about, because this is the very first steps, where it's not even being given to an individual who has Duchenne. It's in humans who don't have the disease, just trying to figure out doses and circulation and levels. So if I give you 10 grams of the medication, how much is in your blood? How long does it stay there? Where does it go? And then you can start to get indications of, is it working on pathways that you're interested in? And then the phase 2A, 2B is where the community starts to hear more about it. And these are the first times that this drug is tried in people who have the disorder. And the questions that we need to answer there is it safe? And is there evidence that it's working? And this is where we employ those biomarkers that are an outcome measure, as well as the outcome measures that we've learned from natural history, to begin to see if we're seeing an indication of any effect. And it's also a good way to, in these trials, to see how are we doing with these outcome measures. Are the different centers that are doing the study performing them similarly? Are we getting the same type of results from different centers? And is this a good measure? Is it something that's actually reflecting the disease process? We can see in this smaller, shorter phase 2, where there's fewer people involved, before the time is spent on launching a larger study. And then the phase 3 study is where we're really asking the definitive question, does it work? It's a large-scale study. Typically, these are randomized, double-blinded, placebo-controlled studies, although we'll talk about the design of some other studies. It doesn't include everyone in the population in the study. Often it's limited to a subpopulation of a particular age or a particular functional status, in order to be able to see a change over the course of the trial, which may be a fair amount of time investment. It may be up to a year long. The exclusion criteria are generally picked for safety. So if one has heart failure that's not in balance, you might not be included in the study, or if you have some other health problem, they may exclude you because of concern that you might have a complication while you're taking the medication. And the design allows for many outcome measures. So if you're in a study, you'll notice that you do a lot of different outcome measures during your visit. You might have blood drawn, you might have a biopsy done, you might have imaging done, you'll do all kinds of functional tests, walking down the hall, up and down stairs, picking up cups and flipping them over. But there will only be one that is the primary outcome measure. This is the one upon which the statistics depend to demonstrate whether this is working or not. You have to enroll enough people to statistically see a change, because everyone is a little different, has a little bit of difference in where they're starting from, and change over time. So it needs to be a large enough N, and this is called sample size calculation, to have any hope of statistically proving that something is working. And then you need to have a study that's long enough in order to demonstrate change. So if, for instance, in that MRI study, the shortest interval that they were able to detect change in muscle was 3 months, it does not make sense to have a study that's only 1 month long. Some studies also incorporate an open label extension, which allows them to continue to follow functional status over time and get more safety data, but also allows the people who were in the placebo arm to have access to and be treated with the drug in question while we're determining if there's efficacy. And then phase 4 is the post-marketing phase. I think this is where there isn't as much done as needs to be in many drugs, but this could potentially be a very valuable source of information. So post-marketing, the medication is used in a much broader population. There is not an inclusion-exclusion criteria as far as age or functional status, but there will probably be some exclusion criteria as far as safety measures if there's a known concern about heart health or some other aspect. But the range of patients being treated with this drug will be much broader. And this is the time, and this is the phase at which more outcome measures can be followed over time, so we can see how much is it helping these other populations, or is it helping? And to raise the question, are there any specific populations for whom having the treatment has not shown any benefit, and so do we need to continue it if we're not seeing change or seeing a biomarker difference? And so I think phase 4 studies, especially in rare diseases where there's small populations that are eligible or would be a target audience for the medication, in order to understand these drugs over time. And so in Duchenne, there's a lot of different aspects of the disease process that are being targeted by different groups, different strategies, different philosophies. And in the future, it will likely be that, and it already is now, that your clinical treatment will involve multiple treatment strategies. And it already does. If you think about it, your treatment strategy includes a physical therapy aspect, including stretching and maintaining strength. It includes your heart health, with over time beta blockers or ACE inhibitors. It includes your respiratory support, and includes steroids as something that's been shown to have benefit. And most clinical trials allow for the use of steroids as an enrollment aspect, although some do not, because as you can see in here, one of the things that is being targeted is trying to find other and better treatments than steroids that might have a better side effect profile. But there's a number of different targets in the process of Duchenne muscular dystrophy that could be targeted. And just to reflect this, there are quite a number of clinical trials ongoing. Several have reached the phase 3 stage, but you can see that there's many more in this phase 1 or 1-2 stage. So just to go over different types of treatment strategies, back 20 years ago, we diagnosed Duchenne based on muscle biopsy and the lack of dystrophin, and we might or might not have known what the underlying mutation was. But there are many different types of mutations that can cause abnormality in dystrophin expression. Some are having duplications where there's a piece of the DNA that's repeated. Another is a deletion, and that accounts for about 60% of cases. Sometimes these deletions can not be a multiple of 3. In other words, it changes the reading frame. Codons are in multiples of 3, so it takes 3 base pairs to spell an amino acid. And if you take out 2 or 1, you shift the sequence in such a way that it will no longer make sense. You can also mutate the sequence so that it stops abruptly, that there's a stop sign that's put in that wouldn't normally be there. And the last is if there is a deletion of a word, but there is some essential information. And we think that in Becker muscular dystrophy, where there is some but not enough dystrophin, that this is the case, that you have some but not enough protein, and that is one of the therapeutic objectives of getting some protein in some of these other mutation types. So that stop sign that I described previously is something that is a therapeutic target. So over 10 years ago, it was demonstrated that a certain antibiotic, gentamicin, can cause read-through of these abnormal stop signs, but don't cause read-through of the normal stop signs. However, gentamicin, while it's a great antibiotic for certain infections, is certainly not something you want to be treated with on a daily basis because of the side effect profile. It can cause deafness, it can cause kidney failure, and so it's not something that one would wish to be on long term. And so screening tests were done in order to find other small molecules that could do the same thing, to run the abnormal stop sign but stop at the normal stop sign. And they found several candidates, one of which is called attemptlarsen, and that has been through clinical trials and showed at some benefit at a lower dose rather than a higher dose. So another example of where having the right dose is extremely important, because the higher dose was no different from placebo. And this has been provisionally approved in the European Union, but here the FDA turned back the United States application, I think wanting more data. And so that is, I think, kind of, I'm not sure where things are in the design of the next trial.
They're
I don't know.
continuing to appeal to the FDA.
Oh, okay. So as you can imagine, these stop signs can be a problem in other diseases too. So if this is a treatment for people with nonsense mutations in Duchenne, it might also be helpful in other diseases as well. And so it's being investigated in a number of other diseases to see if there is proof of principle for benefit in these disorders also. Another type of mutation that's being targeted are exon skipping therapies. So basically, in your DNA, your dystrophin is not all one long sentence prepared with the meaning that it finally has. It has to be cut and pasted into the final form that gets transcribed into a protein. And so I tried to explain that with my favorite sentence, "The big red dog sat and ran," is that there's some chunks in there that you have to get rid of in order to make it a functional sentence. And so these things are splicing events where you've cut and pasted a piece of the initial RNA transcript out in order to get the mature RNA that your cell uses in order to know how to make the protein. you can imagine that if you have a deletion, that you might be able to cut and paste a little bit extra to take that deletion that was out of frame and put it back in frame. And this is the strategy that's being applied in these antisense oligonucleotide analogs. So in the cutting and pasting process, these are designed in order to bind the signals that instruct the cutting and pasting in order to drive it to either include or exclude in this case, a particular exon. So you've lost one due to the mutation that's causing the dystrophinopathy, but if you get rid of another exon, you can put things back into a reading frame. These are chemically designed to be very, very stable and not broken down by the usual things that break down polynucleotides in your body. And they have generally a very long intracellular half-life. And so one of the studies that was looking at skipping exon 51 is a drug called Jacinda, and it was addressing exon 51 skippable deletions, which account for about 13% of all cases of Duchenne. And its phase 3 was completed. It's a drug that's given subcutaneously and either weekly or alternately weekly, and it was almost a year-long study, but it did not show significance in its 6-minute walk test, and the FDA, it was applied to the FDA and it was rejected in January. But it's not the only medication that's addressing the exon 51. There's another drug called attemptlarsen, which was designed by Sarepta, and it is having an ongoing phase 3, which is a little bit of a unique study design. So rather than being a placebo-controlled double-blinded study, it's using an open-label design where the control group, instead of being a placebo arm, are children who have a deletion that is not exon 51 skippable as a comparison group. This medication is a weekly IV infusion, and the original study was between the 7 and 16-year age group, and it did involve a muscle biopsy, and this is ongoing, and it's a 48-week-long study. There's an additional study starting to, that is looking at the younger age group as well. But recently, it was submitted to the FDA based on the phase 2 data of 12 individuals total. And while the committee initially recommended rejection in April, very recently, actually that should be September, recommended provisional approval. But there were some additional recommendations that went along with that that will probably involve some gathering additional data and more safety data. And we don't know what all of this is yet, but hopefully in the next week or two, we will know more about what the path forward is for the attemptlarsen medication. So not only can exon 51 be skipped, you can imagine there are other exons that might benefit other children for skipping. And these studies will be recruiting very soon. And we are one of the sites for this study. It does involve muscle biopsies, and there is a planned open label extension. But I think a lot of people are also interested in non-mutation specific approaches. In other words, are there medications that can benefit everyone who has Duchenne, regardless of what their mutation is? And so some of the things that are being addressed are there ways that muscle can be encouraged to rebuild? Is there a way to decrease inflammation and the contribution to damage that does? And is there a way of making a better steroid that has fewer side effects? And so one of the studies that we're doing here is on myostatin, which is an interesting protein. So in muscle development and maintenance, there are different they're like hormonal signals that regulate muscle size. And one of the ones that inhibits muscle size is called myostatin, myo being muscle, statin meaning to stop. And it's actually a natural signal in all of our bodies, but there are certain animals where this was noticed that it was missing, and one is Belgian blue cows that are what are called double-muscled. They're very muscular. And then I think there's a strain of whippets, and I'm not a dog expert, but whippets are generally these very thin, graceful, almost look like greyhound dogs, which is not what this individual dog looks like. But they are seen as some mutations, and there's actually, I think, a family that was described in the literature where there was a child who had a myostatin mutation who had, greater than expected muscle bulk as a young child. And so we are participating in a study of a myostatin inhibitor drug. It's in the phase 1 and 2 phase. It is double-blinded and placebo-controlled, so I don't know who is getting what. It's a weekly subcutaneous injection, and I believe we are still recruiting. No, we are not recruiting. Okay, I'm getting the wave. Okay. It does require that you're able to do some of these outcome measures, such as doing a 6-minute walk and walking up 4 stairs. And it's a fairly short study of 24 weeks. Another way of addressing muscle function is to try to upregulate utrophin. So utrophin has a similar structure and function to dystrophin, and when a muscle is regenerating and repairing, it is already naturally upregulated. And it can help muscles compensate. If you look in mice that have an increase in the normal upregulation of utrophin, is that their muscles do better. And this is a study of a utrophin upregulating molecule that is being done in, I believe, Italy. It is an oral medication, so not an infusion. It also involves biopsies, but they are also using an MRI as an outcome measure as well. And that study was recently completed and published. It looks like the safety profile is very good. But one thing that was interesting when we're looking at the pharmacokinetics being how does the drug get moved around and metabolized in your body, is that in the individuals who were on the study, the levels of the drug in their blood was much more variable than it was in the phase 1 normal controls, and it was lower. And so the question was, I think remains, is, what is the right dose going forward for the next phase? But interestingly, we talked about CK as not a terrific biomarker in Duchenne, but they did see that CK was decreased in the treatment arm in this study. So this is a strategy that is going to, I think, move on to the next phase. Another approach is to upregulate signals that enhance muscle growth and regeneration, and one of those signals is called Follistatin. One way of doing this is called a histone deacetylase inhibitor. So if you think of DNA as being an extremely long string that your cells need to manage and regulate and make accessible without it being a complete inaccessible tangle, they're actually spooled around these groups of proteins called histones. And then those histones get labeled so that DNA is more or less accessible to the machinery in the cell in order to transcribe or to get to those genes so it can use it. And HDACs help loosen up those spools and spindles so that the DNA is more accessible. And by doing that, a variety of proteins, not only follistatin, can be upregulated. And this is an approach that is being used not only in dystrophinopathies, but in other disorders too. But when this was tested in mice, the Duchenne model in mice, it enhanced muscle growth. And it has completed its phase 2 study in Italy. I I don't know what the results of that showed, but the phase 3 is in the planning phases. And so that is the extent of what I have. I did get some interesting questions in the break. I was just going to talk to the rest of the group about it a little bit too, about a lot of people are interested in some approaches that have hit the news a lot lately, such as CRISPR. So I don't have any slides on it, but CRISPR is a technique in which you can permanently modify the DNA in your genome, in the nucleus, with one treatment event. So literally, it is a molecular scissors, an endonuclease, that can cut the DNA. And it cuts the DNA based on a sequence that's called a guide RNA that finds a specific sequence, something that says this is this part of dystrophin. And so that guide RNA binds there and directs where these molecular scissors should cut. You can also introduce, once the DNA is cut, a sequence to put into that gap. Or you can have no instructions to insert in there, but cut at 2 locations and cause an exon skip, much like the antisense oligonucleotides do that I described earlier. There is a group that has tried this technique in mice, mouse models of MDX. They get the CRISPR system into the cells using a virus called AAV. It took 2 separate viruses to get everything into the cell that was needed. The idea was to cut in 2 places so that you could cut out a chunk of the DNA to put things back in the reading frame that's necessary. And they were able to demonstrate that they could accomplish this. But it was at a fairly low level. So it was only a few % efficiency where everything aligned correctly. You got both viruses in the cell and you got things cut, and then the dystrophin was expressed. One thing that's interesting and important to know about CRISPR is that it will cut based on the guide RNA accuracy, but it can also cut in other places too. And so if CRISPR doesn't have a guide RNA, or if the guide RNA is a little bit non-specific, in other words, it can bind in more than one place, you could cut in more than one place. You could cut areas that are not the dystrophin. And when that happens, you can end up with 1 or 2 base pairs missing. And if you think about the reading frame that we talked about before, having 3 base pairs gives you a shorter sentence, but 1 or 2 will put things completely out of frame. So you want to have a system where there's not a problem with this off-target effect. And so not only are they designing techniques to look at dystrophinopathies with CRISPR, the whole field is looking at CRISPR as a tool of ways of making it much more specific. So it might be the CRISPR technology that's out there now is not as strict as it needs to be used in human trials. But there are a number of different molecules that are being evolved, designed to have that level of specificity. But I think one thing that all of the regulatory groups are going to require is to look for and know if there are these off-target effects. But it is a pretty exciting prospect to be able to correct a germline mutation permanently with one treatment. But right now, they're still working on getting it to work in mice. So I just figured I'd sum it up for everyone else here too. So.